What happens to the sarcomere when it's stretched or contracted? Well, first of all, the A band always stays the same length, no matter if it's being stretched or contracted or if it's in a resting state. That means that's the length of the myosin filaments, the thick filaments, always stay the same. However, the I band appears to get longer when it's in a stretched configuration because the sarcomere itself is getting longer and there's less overlap between the myosin and actin. And in the contracted state, it appears to be shorter because there's more overlap. The H band also changes. It becomes larger in a stretched configuration and smaller in the contracted state. However, the A band, as I said previously, doesn't change. At the EM level, you can really see the details of all of these different bands and lines. For instance, the sarcomere goes from Z line to Z line, and the H band, which is pretty small in this particular micrograph, can be seen there at the center. It's a lighter area in the middle of that A band, and it's bisected by the small M line, which has the accessory proteins that bind everything together. This is how that looks diagrammatically, and also there are a number of accessory proteins that anchor the thick and thin filaments into their positions. For instance, the actin is anchored by alpha-actinin at the Z line. There's also titin, which helps organize the interface between the thick and thin filaments. There are two proteins, myomycin and C protein, in the M line, which help anchor and organize the thick filaments. So at lower magnification, you can see some nice features that we saw histologically, and some things that are difficult to find histologically. So here you can see portions of 3 different muscle fibers. There's one here, another one from here to here, and the third one starting here and going off the edge of the image. Between those, you can see the evidence of the endomysium, that delicate connective tissue layer between muscle fibers. Within each one of these large muscle fibers, you can see numerous myofibrils, each one of them ensheathed by sarcoplasmic reticulum. And you can also see the sarcomeres, which are joined end to end to end along the length. So recall the T-tubules from our prior study of striated muscle. You also have T-tubules in skeletal muscle, and these are closely associated with the terminal cisternae of the sarcoplasmic reticulum, which is where the calcium ions are stored. The T-tubule system are invaginations of that sarcolemma, which bring the change in membrane electrical potential into the middle of the cell, where it can interact with those cisterna of the SER. This allows for a very quick contraction of the myofibrils throughout the cell, in spite of their sometimes very large diameter. And because you have 2 cisternae of the sarcoplasmic reticula flanking each T-tubule, you have then a triad. And that triad is located in a very specific place in skeletal muscle at the junction of the A and I bands. Now let's look at the neuromuscular junction. Another name for the neuromuscular junction is the motor end plate. Those terms are used pretty much interchangeably. What is it? It's a synapse between a motor neuron axon terminal and a muscle cell. It uses or utilizes the neurotransmitter acetylcholine, and that neural activity is responsible for a trophic effect on the muscle cell, without which the muscle will in fact atrophy. So what is a motor unit? A motor unit consists of a motor neuron whose cell body resides in the spinal cord, and all of the muscle fibers that that neuron innervates. You can have small motor units, maybe one neuron innervating about 10 muscle fibers, and that's associated with fine motor control. Or you can have very large motor units where one neuron is innervating up to about 2,000 muscle fibers. And that situation can be seen in gross postural control. So this neuromuscular junction has very specific structural features. First of all, the axon is covered by Schwann cell cytoplasm all the way down its length, but not where it contacts the muscle fiber. So it pretty much ends its covering at that point where the muscle and axon meet. That axon terminal interfaces with the muscle, has a lot of energy-producing mitochondria as well as numerous synaptic vesicles inside of it. And the muscle, which it's contacting, has very specific structure. The sarcolemma of that muscle cell is thrown into these folds called junctional folds, which increases the surface area, and that allows for more acetylcholine receptors within that membrane. This is an EM showing that exact situation. At the very top, you can see a very small amount of Schwann cell cytoplasm, which is only covering the exposed part of the axon terminal. Then you can see the axon terminal itself with numerous synaptic vesicles and mitochondria in it, and you can see all of the junctional folds of the muscle cell which it's contacting. Interesting thing here is you can see very clearly that synaptic cleft. So what happens here? The synaptic vesicles fuse with the membrane of the axon terminal, and their contents is released into the synaptic cleft. It diffuses across that cleft and binds to the receptors in the junctional folds of the sarcolemma. There are several diseases of the neuromuscular junction, specifically myasthenia gravis, Lambert-Eaton syndrome, and botulism toxicity. Nerve stimulation at the neuromuscular junction is responsible for that trophic effect on the target muscle cell. And if you lose this effect, you can have something called denervation atrophy, where the peripheral nerve injury or lack of peripheral nerve stimulation to the muscle leads to atrophy of that muscle. Let's talk about the muscle spindle. What is this? The muscle spindle is a sensory receptor or proprioceptor that detects the amount of stretch in a particular muscle. These are afferent sensory nerve fibers which are wrapped around spindle cells. Now, spindle cells are specialized or modified muscle cells, and when they go through stretch, it stimulates those sensory fibers, and that sends a signal back to the CNS that tells you how much stretch is happening, and it also allows you to know your position in space. That's proprioception. So this is the basis for the deep tendon reflex or stretch reflex. So the number of muscle fibers is determined prenatally and basically remains constant throughout life. The muscle can grow in two ways. As you are growing as a child, you add sarcomeres onto the end of myofibrils so that the muscle can increase in length as the person grows. But if you want to build muscle like bodybuilders, what you're talking about there is circumferential muscle growth. And that occurs via something called hypertrophy. In hypertrophy, you increase the size of the myofibril diameter and you increase the number of myofibrils per muscle fiber. So the skeletal muscle is adaptive throughout life, and it occurs by changes in both the fiber size and the fiber type. So you can adapt a muscle for longer contraction or faster contraction if you're, say, a sprinter or something like that. So the concept of muscle plasticity means that if you don't use a muscle, or if you have denervation, or maybe you have a muscle or an entire limb that's immobilized after an injury, the muscles there will atrophy from lack of use or from denervation if there's nerve damage. But the opposite occurs when you have hypertrophy. So if you're building muscle, you can increase both the diameter and the number of myofibrils. So it's a use it or lose it situation. What other things can affect hypertrophy or atrophy? Well, age, first of all. You have a reduced ability to hypertrophy with advancing age. And in fact, because a lot of people move a lot less when they're older, they often undergo a lot of atrophy with age. Your nutrition has a big effect on whether you can build muscle or whether you're going to lose muscle. And your hormone state as well. So this is why men can bodybuild much more successfully than women, because they have more testosterone, which is an important component of muscle building. Other hormones, such as growth hormone, FGF, and IGF, promote hypertrophy as well, whereas cortisol causes muscle breakdown. Skeletal muscle does, in fact, have some potential to regenerate new fibers. There are specific myoblastic cells called satellite cells, which are located outside the sarcolemma of muscle fibers, but they are contained within the basal lamina. Every muscle cell is, in effect, encircled by a basal lamina called an external lamina in the case of muscle. And if there's a satellite cell present, they can become activated, re-enter the cell cycle, and proliferate to form myoblasts, which fuse to form myotubes and ultimately new muscle fibers. Very similar to the process that occurs prenatally. However, if that basal lamina is disrupted when the area is injured, then the fibroblasts come in and repair the injury with scar tissue. So you don't get new muscle in that case. So it has to be a very specific situation in order to regenerate myofibers. So there are congenital muscle diseases caused by genetic mutations in the muscle transmembrane protein dystrophin glycoprotein complex. Dystrophin is something that helps anchor the muscle fibers, primarily in skeletal and cardiac muscle. And when you have a mutation in this dystrophin gene, you end up with Duchenne's and Becker's muscular dystrophies, both of which are X-linked recessive disorders. And in these situations, the muscle fibers begin to degenerate and die, and then they end up being replaced by connective tissue and fatty tissue. And that can be seen in this micrograph. These micrographs demonstrate the distribution of dystrophin in a healthy, normal muscle on the left and in a boy with DMD on the right. And you can see that the dystrophin is really just beneath the cell membrane, and every single muscle fiber has it in a healthy individual, so it's anchored properly. However, in the DMD specimen, there's very little dystrophin, and you also see hypertrophy of the remaining muscle fibers. And this is where you have the situation where adipose and fibrous tissue comes in and takes the place of those degenerated muscle fibers.